Okay, all right. So last class we talked about a lot about apoptosis and neuro and a little bit about neurotrophins. So today we'll be only concentrate about neurotrophins. No more apoptosis, and because we have talked a lot of this, but we will have some slides for the apoptosis also, kind of recap. So neurotrophins and neurotropic receptor are apoptotic regulators. So they inhibits apoptosis, and I'll be covering some of these neurotrophins and their receptor. So we have four neurotrophins like neurotrophin 3, neurotrophin 1, BDNF, and NGF, nerve growth factor. So, so for these neurotrophins, you have a specific receptor. These are called neurotropic receptor. So these neurotrophins, either BDNF or NT3 or NGF, they're synthesized as a precursor molecule, like precursor proteins. So you do not have such activity. So these are called as pro. For the BDNF, you have the pro BDNF, and so you have pro NT3 and pro NGF. And each protein has their effect on their receptor. Specifically, their effect on the receptor depends on the status of these neurotrophins. Like specifically, the pro BDNF acts on this receptor like P75NTR. So BDNF will not act on this. The pro BDNF will act on this, and this act as a pro apoptotic molecules. So because of when you have binding of this, so I'll talk about in the future slides where I'll show that this receptor has an important role for neuronal survival, for selection of the target neuron and all these things. We'll talk about this when we'll go through this lecture. So we'll cover up this, the role of P75 NTR. So just remember that pro BDNF binds to P75 NTR, and it is a pro apoptotic regulator which which basically regulates growth cone collapse and also synaptic depression. But these processed molecules binds to this receptor. Like for BDNF, you have TRKB, tyrosine receptor kinase B receptor, and for the NT3 can bind either of these. Like you can bind to TRKA or TRKB. We will talk about this. And after binding of this the molecule to the receptor, you have receptor dimerization. Basically, two molecules of BDNF binds to this receptor and they dimerize. They can phosphorylate this intracellular domain, which is tyrosine receptor kinase domain. You have phosphotyrosine residue, which I'll be talking in the coming slide. So basically, their interaction, because of the interaction, you have activation of receptor protein tyrosine kinases. There are like three I have shown here, and you have because of this, you have neuronal survival, neuronal neurite growth, synaptic potentiation, neuronal differentiation, all of these. So here showing typical structure of receptor tyrosine kinases, the neurotrophin receptor, TRKA. So basically, it has these are the receptor, and this is the extracellular domain of this receptor. These are the transmembrane domain. This is the intracellular domains. So when the basically two neurotrophins binds to this receptor, then after that the ligand causes receptor to dimerize. Okay, and so after dimerization, you have then you have activation of this intracellular domain where they phosphorylate basically each other at the phosphotyrosine residue. So you have a tyrosine residue here, and also another molecule which is phospholipase C can also phosphorylate these two intracellular domains. And after activation, so after because of phosphorylation, you have recruitment of the adapter, like effector protein, not adapter, effector proteins, which is AKT and ERK, which can further initiate the downstream signalings. Okay, so here I have listed the neurotrophins and the receptor. So for NGF, you have tyrosine receptor kinase A. For BDNF and NT4, you have tyrosine receptor kinase B. Then you have for NT3, you have a tyrosine receptor kinase C. And these are basically neurotrophins and their receptor. And some of these other molecules which can also cause, they are not neurotrophins, they are trophic molecules. They can also cause neuronal growth. Like, these are called as, they're known to be trophic. Like they can also cause neuronal growth. These are FGF ligands which can bind to the FGF receptor. You have insulin. It can bind to the insulin growth receptor. All of these, like FGF receptor, insulin growth receptor. You have CNTF, like family member like ciliary neurotropic factor. You have a GDNF, which is glial derived neurotropic factors. So they also cause neuronal growth. So these are shown here, like all of these can cause neuronal growth. So here there are some examples which can cause which can work like neurotrophins. So they belongs to GDNF family ligands. So the GDNF is the glial derived neurotropic factor. So here I have listed 4 of these family member. One is GDNF, which is like glial derived neurotropic factor. You have NRTN, which is neurotrophin-like factor. You have artemin-like factor, percephin-like factor. Like these are the artemin, percephin, all these things. So they have these are the ligands, and these are the receptor for this. Like you have the for GDNF. So all these GDNF family member have receptor like GFR alpha. So one subunit like receptor. So after binding of this, you have activation of this, phosphotyrosine kinase like domain present in the in the cytoplasmic side, which can phosphorylate and again can activate downstream signaling like ERK and AKT processes. So it is shown here. So here you can see that the dimerize, and basically the dimerization of this receptor depends on the red protein, which is which causes it's a receptor type of protein, tyrosine kinase like protein, which causes dimerization and phosphorylation in the intracellular domain. And you have again further downstream signaling cascade for the activation of ERK and AKT. Okay, so phosphorylation and activation of ERK and AKT. So then we'll talk about another trophic factor. This is called a ciliary neurotropic factor, CNTF. Can also causes neuronal growth. So they are not neurotrophins. They are neurotrophin-like factor. Okay, they can also cause neuronal growth. So here showing CNTF receptor. So you have, it is a trimeric receptor which has LIF receptor and also another receptor which is CNTF receptor. So the after binding and of this ciliary neurotropic transmission factor, it again activate downstream signaling cascade, basically, and JAK family tyrosine kinase effector. Basically, it can activate these JAK family tyrosine kinases, which can further give signal to this effector like AKT, ERK, or START. So START again is a transcription factor. Which after phosphorylation START, it enters into the nucleus. Phosphorylated START enters into nucleus. So it is signal transducer and activator of transcription, that is START. So after entering to this nucleus, it binds to this DNA, and it causes the transcription of the genes required for the neuronal growth. Okay, which is shown here. So we talk about some of these neurotrophins and their receptor, like for like BDNF or NT3 or NT1, and the receptor like neurotropic, all these receptor like A type or B type. And we also talk about some of the trophic factor, which are not neurotrophins. They can also cause neuronal growth, like ciliary neurotropic transcription factor, or glial derived ciliary neurotropic factor, or glial derived neurotropic factor. All these things we talked about. So now we're talking about specificity of the trophic interaction, how this specificity is achieved, which receptor has which neurotrophins, and their location, where it is present in the body or in the tissue. So here this is showing that the specificity in trophic interaction in context to the in the skin, which type of receptors are present, like in the neurons which is going to the skin, which type of receptor are present in that neuron, and which type of neurotrophins present in the skin, all these things. So you have specific interaction with their specific neurotrophins. So here, these are the neurons, the dorsal ganglion, dorsal root ganglion neurons. It is innervating to the muscle, or it is going to the skin. So in the muscle spindle, basically it has NT3, neurotrophins. So you have receptor present in the neuron, and you have these molecules like neurotrophins present in the targets. Here the target is the muscle bundle, okay, muscle spindle. So here in the muscle spindle you have NT3 neurotrophins, and basically this neuron going to here in the muscle spindle, or in the skin, in the hair follicle you have NT4 neurotrophins, and in here in the Merkel's disc in the skin. So here in the skin this is the epidermal layer, and you can see that you have enfolding in the skin, and this is called as Merkel's disc, the enfolding. So you have BDNF neurotrophins in the here. So you have again the innervation of this neuron going to to the skin. So you have different types of neurotrophins in a different location. In skin itself, have hair follicle, you have NT4, then Merkel's disc, you have BDNF, you have muscle spindle, have NT3. So you have this specific interaction with their receptor, with their targets. Okay. Similarly, in the peripheral nervous system, like you have specific type of receptor and neurotrophins interaction. So in the muscle, you have in the peripheral nervous system, you have NT3 and GDNF. In the skin, that I've shown that this is the peripheral nerve. So you have NGF, nerve growth factor. So so showing that you have specificity in their interaction. So muscle will have like NT3, it will not express NGF. So that's why you have specific neuron from the dorsal root ganglia will innervate to the muscle. Okay, so because you have a specific receptor for NT3 or a specific receptor for BDNF. Okay, so you have this type of interaction. So here I'm showing kind of summarizing the things, like you have different receptor, you have different neurotrophins. So skin, in the skin you have many neurotrophins, like you have an NT4, you have NT3, or you have BDNF. Merkel cells, you have BDNF, you have D hair afferent, you have NT4. So all these type of diversity are there. So again, in the in the neurons, the neurons which is also innervating skin have a different type of receptor. You have either TRK or P75NTR or GFR alpha 2. So you have different types of receptor itself present in that neuron, and different type of neurotrophins present in the target. The target here is the skin. Okay, so here showing the diversity, like in the enteric neuron you have this type of receptor, like GFR alpha or C ret. So in the muscle, again, you can see you have different types of neurotrophins, like GDNF, NT3, BDNF, NT4, all these things. So thing is that in the CNS neurons, in the central nervous system neuron, they expresses multiple receptor, and they can expose to the multiple neurotrophic factor. So if you lose one of the neurotrophic factor or one of the neurotrophic receptor, you do not see any you have very little effect, because you have multiple of these. You have multiple receptor in the central nervous system, or in in the target you have multiple neurotrophins. Okay, that is kind of highlighting here. But when they mature, whatever I talked about in the diversity, they are in the very young stage of the development. So in the young stage, you express multiple receptor, multiple neurotrophins. But when they mature, they lose their targets. But then how these neurons will survive? So in that case, another mechanism kind of plays an important role, where even if you don't have enough neurotrophins or a neurotrophic receptor, but still the neuron can survive, because it can the mature neuron has kind of neuronal activity. It can depolarize, and their membrane, which helps in the calcium entry, and the calcium can helps in fusion of the synaptic vesicle. It can maintain the activity. And also you have down regulation of apoptotic machinery. So you have down regulation of backs, you have down regulation of back and backs. Also you have up regulation of BCL2 kind of BCL2 is anti-apoptotic, or you have you have also regulation of IAP, which is an inhibitor for caspases. So IAP inhibits caspases. So you have expression of IAP protein so that neuron can survive, because so that is how the mature neuron survives. So in the mature neuron, what happens in the young neuron, you have many receptor, many neurotrophins. Okay, that's why you have a lot of combinations. But when the neuron mature, they lose their either the receptor or in the neurotrophins present in the target tissue. But then how the neuron, some of the neuron will survive? Because they maintain activity, because they can maintain activity by depolarizing their membrane, and also they can upregulate the anti-apoptotic pathway so that the cells will not undergo apoptosis. They can downregulate the apoptotic genes like BAX and BAC so that apoptosis will be inhibited. Then you will have survival of the neurons. So that is how the neurons survive in the mature stage. Okay. So we'll talk more about the TRK signaling transduction pathway, and so we'll talk more about this in more detail, how the receptor and the ligand, the neurotrophins interacts, and how they can actually initiate downstream signaling cascade. So here I mentioned that two neurotrophins bind to the receptor, then they dimerize. So two NGF molecule bind here, then there is the conformational changes. Then these are the then you have in the intracellular side, you have this kinase domain which phosphorylate at the phosphotyrosine residue. The phosphorylation is they can phosphorylate each other. They are autophosphorylation, and also they can phosphorylate by phospholipase C. So after phosphorylation, the intracellular domain get activated and they can recruit their effector. You have the effector kind like AKT protein or PKB, protein kinase B. Also, you have activation of ERK and MAP kinase. These are also effector. Then also you have RAC and RO recruitment, which can actually inhibit the spine formation. So here the function of effector is like here I mentioned this function of the effector, like protein kinase B or AKT, they helps in survival and growth. And for the differentiation, you need activation of ERK and MAP kinase. And for growth cone collapse, you have activation of RO. Okay, so that is how the neurotrophins can actually regulate cell differentiation, cell survival, and also growth cone collapse. Okay, so we talked about this, and independently this phospholipase C can also initiate calcium signaling, because in the mature neuron, calcium signaling is important because you have influx of calcium to the presynaptic terminal, where in the mature neuron you do not have enough neurotrophins or neurotrophic receptor. This calcium signaling can also maintain this neuron in a healthy state, because you can fuse the synaptic vesicle, you can maintain the activity. So here showing that function of different adapter or the effector complexes. So we kind of partially covered about this apoptosis in the context of neurotrophic factor signaling in the mitochondria. So here I want to highlight again how the neurotrophic signaling can regulate the apoptosis. So here, this is the mitochondria, and here is the neurotrophins. We are talking about the NGF. So when NGF binds to this receptor, neurotrophic receptor, what happens is you have dimerization, you have autophosphorylation in the intracellular domain, then you have a recruitment of the effector. And also you have activation of other GTPases like RAS GTPases or RAP GTPases, which can basically activate the ERK. So this is how the ERK is getting activated. And also this AKT or protein kinase B get activated by phosphoinositide 3 kinase. Okay, when this get activated, they actually protein kinase B phosphorylate bad protein. Basically, bad is a pro-apoptotic regulator. If you have BAD is recruited to this mitochondrial membrane, what happens is it can form the pores, it can permeabilize the membrane, outer membrane. Then you have release of cytochrome C from the intracellular space to the cytoplasm, which can make complex with PAF1. Then you have activation of caspases and cleavage of a lot of components. So the BAD can do that. But to inhibit the activity of BAD, what happens is the protein kinase B phosphorylate, and because of phosphorylation, the BAD remain sequestered in the cytoplasm. So that's how the neurotrophic factor signaling can inhibit apoptosis. Okay. So here showing that how it can also transcribe or translate anti-apoptotic protein BCL2. So here the RAS and RAP. Here the function of PI3K kinase is activation of AKT and protein kinase B and a sequestration of this BAD protein. But here the RAS and RAP, basically what that does is they can activate activate the ERK kinases. After activation of ERK kinases, it can translocate to the nucleus and again further activate the CREB transcription factor. And CREB transcription factor binds to this DNA and transcribe this BCL2 mRNA. Then the BCL2 mRNA get translated to form BCL2 protein, which is anti-apoptotic, and it can recruit it to the membrane. It can stop the release of cytochrome C from the intermembrane space. So that's how you have survival. You have inhibition of apoptosis. Okay, so showing that how TRK tyrosine receptor kinase signaling basically regulates apoptosis. Then the next question is the timing and location. So what time, you have different situation. Sometimes you need transient activity, and sometimes sustained activity. Sustained activities means you need kind of longer signaling cascade. Transient means suppose you need immediately. Okay, so here showing that two different modes of the receptor tyrosine kinase activation and regulations. So here for the transient activities, certain activities, what happens is when the NGF bind to this receptor like TRKA receptor, which is a neurotrophin receptor, the NGF is neurotrophin, binds to it phosphorylated, there will be autophosphorylation in this intracellular domain. Then you have activation of RAS. RAS again is a GTPases, which can activate RAP, which can activate MEK1 and 2 and ERK1 and 2, which causes the transient activity. It can immediately. But for sustained activities, you have activation of RAP1. For transient activity, you have activation of RAS. For sustained activities, you have activation of RAP. Then RAP can again activate BRAF, then MEK1 and MEK1 and 2 and ERK1 and 2, which can we can sustain the activity for longer period of times. Or what happens sometimes is you have endocytosis of this entire complex like neurotrophin and its receptor to the cytoplasm. Then after that you have recruitment of a RAP1, which is a GTP bound. In the GTP bound state, it can activate RAP, which can further initiate the downstream signaling cascade for the sustained activities. So just remember that for transient activity you need activation of a RAS protein, and for sustained activities you need activation of RAF protein, mostly the RAP1 and RAF both. Okay, so that is how you have regulation of neurotrophins in the cells, like for the transient and for the sustained activities. So then the next question is actually where you have this receptor present in the neuron, the neurotrophin receptor, the TRK, TRKB or TRKC, whether it is present in the cell body or it is present in the axon terminal. Okay, then we want to know that where it is present, the location. So what happens is here in the experiment, it has been seen that basically neurotrophin receptor are present in the axon terminal, and after binding of this neurotrophin to this neurotrophin receptor in the axon terminal, you have endocytosis for the transient sustained activities. You have endocytosis of the entire complex to the axon terminal. Then you have retrograde movement to the cytoplasm, to the cell body, because cell body is far away from the exon terminal, and there you have expression of a gene. So that's how they have kind of hypothesized and proved that. So what they have done here? So here this is the cell body, and here is the exon terminal, and here they kind of inactivated these microtubules by they applied certain depolymerizing drug which can depolymerize the microtubules, and microtubules helps in transport of vesicles, basically different kind of vesicles from exon terminal to the cell body. Okay, that is called retrograde transport. So when you depolymerize here, sometimes in the somewhere in the middle, what happens is whatever the vesicle is transported will through this exon to the cell body will be inhibited. So they found that basically those vesicles, after interaction with neurotrophins and with its receptor, they get endocytosed, they stop there, they could not transport to the cell body to initiate further downstream signaling cascade for cell survival or neuronal growth. So here they have done this kind of experiment, and further I will show kind of in the next slide. So here they have done this experiment to show that retrograde transport of neurotrophin is important for the cell survival, for the cell neurite growth. So here they have this three-chambered having the neuronal culture. So in each chamber you have that NGF, like UL1 you have NGF, and the neurotrophins UL2 and UL3. So in all this situation you have growth of the neurons. Okay, but when you have removed the NGF from UL1 and UL2, you can see that you do not see such growth of the neurons. So this I can kind of diagrammatically show that the mechanism of this, how the neurotrophins regulating this expression and survival of these neurons. So here what happens? So they basically with different techniques like imaging technique and different methods, like here they depolymerize these microtubules. They found that the neurotrophin receptor are present in this exon terminal. Basically here these are the neurotrophin receptor, and when the neurotrophins binds to this receptor, they get phosphorylated. And there are two modes of activities, like transient. For transient, they can activate RAS, and for sustained, they can activate RAP. And also there is endocytosis of this entire complex. So like neurotrophins and its receptor to its exon terminal, then that get transported through this exon, retrograde transport, and it enters into the cell body. Here in the cell body, what it does is it can activate, because since these are phosphorylated, it can activate AKT and ERK signaling pathway, which is required for the cell survival. It can also activate the CREB transcription factor, which can kind of regulate the expression of downstream genes. So that is what they have seen. So but in the exon terminal, the role of AKT, because you have activation in both the sides. So here also you have activation of AKT and ERK1 in the exon terminal. So that helps in the exon extension. But the activation of AKT and ERK in the cell body helps in the survival of the neurons. That is what they have found. So there is a transport, retrograde transport of this complex neurotrophins and its receptor to the cell body for the survival and maintenance of the neuron. Okay. So in the very beginning slide, I talked about another neurotrophin receptor, the P75NTR, and I did not, kind of highlighted that. So here we have, so this is the first slide I talked about, the neurotrophin receptor P75NTR. Basically, it binds to this, probe, neurotrophins like pro-BDNF, and here they have seen that, and their binding depends on the binding to the P75, NTR75 depends on the type of neurotrophins. So basically the mature, where you have synthesis of pro-neurotrophins, and the pro-neurotrophins basically get processed by enzyme present in the in the matrix metalloprotease that is present in the extracellular matrix of the cell, basically that act on these pro-neurotropins. And you have here you have kind of cleavage of this, and after processing you generate the mature neurotropins. Basically, the mature neurotropins binds to the TRKA or TRKB or TRKC. The pro-neurotropins binds to this P75 NTR. Okay, so the mature neurotropin doesn't bind here. The pro-neurotropins bind here. So after binding of these pro-neurotropins, it has a death domain which can induce apoptosis. Okay, so when the cell wants to kind of kill itself, they induce this mechanism. So for the apoptosis by this receptor, pro-neurotropin receptor P75 NTR. Okay, so that is how you can actually regulate the activity or survival or the growth of the neurons. So here showing that different signaling cascade got activated after binding of either pro-neurotropins to the P75 NTR or the mature neurotropin to its neurotropic receptor like TRKA, TRKB, and TRKC. So here showing different signaling cascade recruited after binding to the P75. You have a recruitment of other factors here which can helps in cell cycle arrest. The NADE can cause a cell death. You have when you have activation of Rho, it can activate Rho protein, Rho protein, which can depolymerize cytoskeleton, which can kind of inhibits the neurite growth. It can activate the RAC1 proteins. So RAC1 has a dual role. RAC1 can also induce neurites growth. It can also induce retraction of neurites. So here showing the dual role of RAC. So when we have activation of P75 NTR, then you have here the RAC causes the death of the neurons. But in other cases, we have seen that in the previous some of my lecture, I have shown that like RAC1 helps in philopodia formation, like neurite growth. Okay, so these are the signaling cascade which activated when you have binding of the pro-neurotropins to the P75 NTR, and when you have binding of these neurotropins, mature neurotropins to its receptor, TRK receptor like TRKA or TRKB or TRKC, this type of signaling cascade get activated. Like you have activation of PI3K, which can activate AKT. AKT required for cell survival. You have also activation of a RAF or activation of a RAS proteins, so which can lead to the cell survival. Okay, so JNK is showing that role of JNK and ROA in the cell death or cytoskeletal changes. Okay, now in the very beginning of my lecture, I talked about during birth or when you have very early stage of the development, you generate many neurons, but all the neurons they do not survive at the latter stages, because I told that there is always a competition to get the neurotropic factor. The neuron which get neurotropic factor, which wins in the competition, they remain in the latter stage of the development. Other neurons which cannot compete, they die. But why? Then another question arises: why all the neurons? So one hypothesis is that they compete and they win. You can also think about the neurons which get insufficient neurotropic factor. Why don't all the neurons die? So one will assume that you can have loss of all the neurons, right? But why there is survival of some of the neurons? So what is the mechanism? Basically, these neurons, why they are survived? Of course, the like very simple answer is they win in the race. They get the neurotropic neurotropins. That's why they survived. Then what is the mechanism? How they survived? Okay, so we are going to talk about some of this mechanism of this survival who win the race and in the competition. Mostly, I will be taking example of sympathetic ganglia. How some of the sympathetic ganglia they survived in the latter stage of this development. Okay, so in the earlier stage of the development, you do not have neurons. You have progenitor for the neurons. Okay, so these are the progenitor cells, and these are non-neuronal cells. So here they have shown that you have expression of two different kinds of neurotropins. One is neurotropin 3 and another is NGF. Okay, So in the earlier stage of the development, the non-neuronal cells expresses neurotrophin 3, NT3. So NT3 basically expressed in the earlier stage of the development in the non-neuronal cells, which are present adjacent to the ganglia or neuroblast, the progenitor for the neural cells. These are the progenitor for the neural cells. These are the neuroblast. So these cells, non-neuronal cell, expresses NT3. Okay. And these cells, basically, in the earlier stage of the development, as I mentioned, you have different kinds of neurotrophic receptor like TRKA, TRKB, and TRKC. So in the sympathetic ganglia, you have two different types, TRKA and TRKC. Okay. So in the earlier stage of development, these neuroblast expresses TRKC. So here you can see that only you have the expression of TRKC, and these non-neuronal cells express only NT3. Okay. But when they develop, when they develop, kind of entering to the mature stage, okay, so you have expression of other receptor, and these neurotrophins which are present in the non-neuronal cells, they kind of migrate to the along with the axons. So here you can see that these color, you can see that here these are yellow, and they you don't have yellow here in the non-neuronal cells. It migrate along the axons. So, and these axons expresses other receptor along, including TRKC, it expresses TRKA. Okay. What happens is you have expression of TRKA. In the latter stage of the development, TRKC goes down and TRKA, kind of dominated. Okay. And you have, these NT3 basically here, they moves from non-neuronal cell to the axon terminal. But when they become mature, you have loss of TRKC, you have expression of TRKA receptor. Okay. So you have expression of TRKA receptor. Now it finds its target. The target expresses another neurotrophins, which is NGF. So initial stage, you have expression of NT3, then it goes down. Then in the final mature neuron, you have expression of NGF. Okay. And all the neurons are TRKA positive, they are TRKC negative. Okay. Now, in these cells, you have expression of, in the mature stage, you have expression of P75 NTR. Okay. You have expression of P75 NTR. So you have P75 NTR here in all the cells, and you have in the target, you have NGF. Okay. Then here, what happens? What happens here is you have expression of another neurotropic in these cells. In the latter stage, you have expression of another neurotropic factor called as BDNF. Okay. When you have expression of BDNF, where you have only P75 NTR, those cells, when you have expression of all these things, like you have different combination, you have expression of TRKA, you have expression of P75 NTR in the latter stage of the development. But BDNF will bind to the TRKA, BDNF will not bind to the P75 NTR. Okay. So those neurons which have TRKA and P75 NTR having BDNF, they survive. But those neurons will get eliminated in the latter stage because binding of pro BDNF. So pro BDNF basically binds to P75 NTR. Okay. When pro BDNF binds to P75 NTR and P75 NTR, the cell dies. Okay, in the very beginning of my slide, I talked about there are two forms of neurotrophin. These are mature form, and you have the pro form. So pro form bind to the NTR, then the cell dies, and the BDNF bind to the TRK, then cell survives. Okay. So here in the latter stage of the development, what happens is just want to summarize. It is a very complicated slide. So in the earlier stage, you have non-neuronal cells. You have expression of NT3. And you have expression of TRKC in the neuroblast. Then when it develops, when it forms the axons, you have expression of two different types of receptor. One is TRK and TRKC. In the latter stage, TRKC expression goes down. You have a dominance of TRKA, and you have NT3 in the axon along the axons. Now in latter, when it reaches to the target has kind of neurotropic factor, which is BDNF. So the target has BDNF. So when you have BDNF, so in the target has either BDNF or pro BDNF. Okay. The pro BDNF bind to P75 NTR because in the latter stage, they have two different kind of receptor. in these neurons, either TRKA or P75NTR. The TRKA binds to the BDNF and you have cell growth, and also it binds to the pro-BDNF. So when it binds to the pro-BDNF, then these cells dies because you have P75NTR. So in the latter stage, you can see that the cells which have P75NTR and binding of pro-BDNF, they dies. Only these cells survive because you have binding of BDNF to the TRKA. Okay, so that's why that is how you have survival of some of the neurons in the latter stage of the development because of this mechanism, like BDNF and pro-BDNF, P75NTR, and expression of TRKA. Okay, so this is the mechanism that how you have survival of some of the neurons in the latter stage of the development. Yes.
In the previous slide, it said that the neurons, is it that the neurons that bind well to the target release pro-BDNF, causing the death of the other neurons, or is it that the target also releases both BDNF and pro-BDNF?
The target releases both BDNF and pro-BDNF. Okay, and when you have expression of binding of pro-BDNF, then the cell dies.
So just like chance, whichever one BDNF survives.
Yes. Yeah. So this neuron survive because you are binding on BDNF. This neuron dies because you are binding on pro-BDNF. So that's why you have in the development you have survival of only the neurons which get correct neurotrophins. Okay. So that is how shown that. So we'll enter the last section of this topic, like neurotrophic factors are also chemotropic factors. Because here, kind of showing that sympathetic ganglia, 6 sympathetic ganglia, when you added neurotrophins to the sympathetic ganglia, you can see that compared to the control where you have not added any neurotrophins, you see that in the experiment there is a large number of neurites growth. That means they promote neurites growth. We talk about this also in the last class, and here showing that the cells which get the wells which get basically neurotrophins, they show growth of the neurites. So well 1 and well 2, they do not have the neurotrophins, then you do not have the growth, but here you have the growth because you get the neurotrophins. So neurotrophins or nerve growth factor or other neurotrophic factor are required for the maintenance of neuronal survival. Okay, they are also required for the local maintenance of neurites and synapses. So that is just want to show that basically they require for the maintenance of the neurons. Okay, so here showing how NGF, another neurotrophins, nerve growth factor, regulates neurites growth. They can act as a chemoattractive cues for the neurite growth. So here here they have taken these neurites and they have filled this pipette with neurotrophic nerve growth factor, and they have kind of approaches to these neurites. And after 22 minutes, you can see that the neurites attracted to the NGF and the nerve growth factor. Okay, then when they kind of pull the pipette, you can see that the neurites is growing in that direction. After 60 minutes, again when they remove this pipette, you might have release of some neurotropic or neurotrophins to this area, so that it is again following this area even after removal of this pipette, it can following the path. So that means the neurotrophins can be as attractive attractive cues for the neuronal growth. And here I want to show some experiment that have been done by using the dominant negative and constitutive active form of this some of these proteins. So before going detail into the experimental part, I want to highlight kind of what is dominant negative and what is constitutive active form of this, and how they regulate different cellular processes. So basically there are multiple protein G proteins which can bind to the GTP present in the cell. They remain either in the inactive state or active state. So the GTP bound form is inactive state of the protein, G protein, and GTP bound state is the active state. Basically, the exchange of GDP to GTP happens because of this protein, that is guanine exchange nucleotide factor, which can exchange GDP to GTP. So this is the active form. Then you have another protein called as GTPase activating protein, which can remove this phosphate from this GTP, and you have bound up GDP, so that then the cell become inactive. So you have these cycles. Okay, so what happens when you expresses dominant negative form of the, like for example, Rho proteins, which is Rho N19. Basically, what it does is it binds to the gap. Okay, it binds to the gap and preventing them from activating endogenous Rho. So you exogenously you are expressing this dominant negative form, this Rho N19, and in the endogenous you have also have expression of Rho 19 Rho proteins. So when you express the dominant negative form, it can bind to the gap, and which can inhibit the expression of the endogenous Rho. Okay, so that means you can entirely shut down the system. You can have inactive form of this, always in the inactive form of this protein in the system. So that's how you can also deplete the endogenous form of this protein. However, in the constitutive active forms, like Rho V14 mutants, it cannot hydrolyze GTP, so it will always in the activated form. That means it will always give the kind of signaling. So here, constitutive form always signal to this, always initiate signaling. So when you express dominant negative form, you will have expression of the GDP bound forms, which will remain inactive in the system, so that you will not have expression of downstream signaling. But when you express, because it inhibits the exchange of GTP to GDP, because it binds to the gap, the inactivated like the Rho 19 binds to the gap. But when you have expression of this constitutive form, then you have always, the GTPase activating protein cannot hydrolyze GTP, so that you have always bound, the protein always remain in the bound state, the GTP. Okay, so always it gives signals. So they have done certain experiment by using the neurotrophins. So here what they have done is they have taken NGF and they have added to the neurons. So you can see that after addition of the NGF to the neurons, you have more neurites growth. Okay, but when they expresses the dominant negative form, inactive form of this RAS protein, RAF protein, AKT, or DNP85, those are the effector for the neurotrophins, which can regulate the downstream signaling cascade and helps in neuronal maintenance. But when they express the dominant negative form with NGF, you see that you do not have such elaboration like in the control. So in the control, you have more axon growth, but when they have dominant negative form, this effector, you do not have the neuronal growth. Okay, so that means neurotrophins regulates axon growth. Okay, so the control and all these things showing that regulation of this. Then again, I talked about in the very beginning of my class lecture about the semaphorin, how semaphorin causes neuronal growth cone collapse. Okay, so here we want to kind of in the final lecture I am showing the mechanism how semaphorin regulates neuronal growth. So here they have taken this culture and here they have added NGF, NOR growth factor. You can see that you have formation of this dendrites like structure, and you have elaboration in the you have growth of the neuron. But when they have added semaphorin 3F along with NGF, you do not see such elaboration, which is also shown here. In semaphorin, even you have NGF, you do not have elaboration of this neuron, like growth of the neurons. And what is the mechanism? Because the semaphorin basically inhibits the effector for the neurotrophins like AKT and ERK signaling. So when you put semaphorin, it basically inhibits the activation of this effector AKT and ERK. That's why you do not have further downstream signaling and do not have the neuronal growth. But this can be suppressed if you overexpress GAB1. So GAB1 basically suppresses the phenotype, which is caused by semaphorin-induced growth cone collapse. So here, when they overexpress the GAB1 in this culture, where you have semaphorin, you can get the neurites. Okay, so GAB1 basically rescue this phenotype of semaphorin-induced growth cone collapse. Okay, so we have some more slides. So here showing that the role of GTPases for regulation of this growth cone. So I have already talked about basically the RAC1 and the CDC42 helps in philopodia growth, and RhoA activation basically inhibits filopodia growth. Here showing that how RhoA regulates growth cone collapse. We have talked about all these things before. Here showing that how BDNF regulates neuronal growth in the so here in the beats you have the BDNF, and here you have the sympathetic neuron. So here you can see that there is growth of the neuron to the BDNF in the response to the BDNF. BDNF bead, but when we express dominant negative from the rack, you do not have such growth. But when we express constitutive form of this rack, you have more growth. Or dominant negative rho can also causes neuronal growth, because RhoA causes growth cone collapse. But when we express dominant negative from the RhoA become inactivated. That's why you have more neuronal growth. Or you can induce neuronal growth by by culturing with lipophosphatidic acid. So here, okay. So this is showing that how the dominant negative causes neuronal growth. The RhoA dominant negative causes neuronal growth. Okay. So these are all shown here. So these are the kind of key points. The TRK signaling basically promotes axonal growth. Both ERK and AKT, these are the effector for this TRK, neurotrophins required for the neuronal growth. And I have shown that some of these neurotropic-like signaling molecules can also cause neuronal growth. All these things are kind of highlighted here. So then the next question is, basically we found that a neurotropic factor is required for the axon growth, and which work independently of the survival. So we'll clear about this concept by going over these slides. So here the wild type mouse have TRKA and its NGF in the targets. So you have axonal growth because you have the receptor, also you have the NGF, neurotrophins. But here you have knockout receptor, the NGF, sorry, the target NGF, you do not have the neuronal growth, even you have the receptor. And here you have knockout receptor, you have NGF, you do not have the neuronal growth. And this is because in this case, you might be thinking because of activation of certain genes, the BAX gene or BAC gene, which causes apoptosis. So in this situation, you might be thinking there is apoptosis, and that's why you do not have the neuronal growth. But what happens when you knock out NGF, the receptor, or the neurotrophins, or the receptor TRKA, along with the BAX, which is also apoptosis genes. In this case, you do not also see the neuronal growth. Okay, one will expect that you will have neuronal growth, but here you do not see, because here you are assuming that you have expression of BAX, that's why you have apoptosis, you do not have neuronal growth. But here you have knockout everything. But in this situation, one will assume that you will have neuronal growth. But here you are not seeing that, we suggest that this NGF and its receptor, like tyrosine kinase A receptor, required for the axonal growth, independent of survival of the cells. Okay. So here some of the experiment here, like BAX plus or TRK, you can see that you have nice neuronal growth. When you knock out TRK, you reduce neuronal growth, or both of these, showing the you require basically the neurotrophins for the axonal growth. Okay. So here some of these key points showing that neurotropic factor signaling, and it's kind of go over these slides. I have already mentioned this thing. So some of these things also, we talked about that neurotropic factor required for the development of the axon growth just before, some of this before the last slide talked about it's required for the axonal growth. And here we'll be talking about some of these, neurotropic factor, how it regulates, like BDNF regulates neuronal growth. So here showing the same thing, like I talked about how the neurons, some of the neurons survive in the later stage of the development. In the earlier stage, you have many neurons, but later stage you have you have less neurons. How they are survived because of this P75 NTR expression. So it's kind of showing that same thing here. So here showing that in the non-neuronal cell you have expression of NT3. Then you have expression of the receptor TRKC in the ganglia, showing the same thing, kind of recapping the things. So here the same thing I'm showing that how this thing happens, the mechanism. You have expression of everything like NTP and NTR75 or TRK and TRKB. So here showing that different modes of action of NT3 or NGF. So you have three type of neurotrophins I talked about, like NT3 or NT1 or NGF or BDNF, four types. So basically NGF endocytose, but NT3 and basically NT3 doesn't endocytose. It can directly signaling, it can direct signal for the axon growth. But NGF basically it can either endocytose or it can also signal through the through these intracellular domains. But NT3 only, it doesn't endocytose. That is what showing the role of different mode of different neurotrophins. So then last segment, we'll talk about how, in the early stage of the development, you have less, neurites growth, or you have less elaboration of the neurons. But when the animal mature, you have more elaboration. You can see that in the different layers, like 3 months old, you have much complex than the newborn. Even in the 2 years old baby, you have more elaboration of this neuron. You have more kind of complex interactions. So how this thing happens? It happens because of the production of the neurotrophins. And here I'll be talking about the last segment, how BDNF plays an important role in neurites growth. So here, this is the control, 27-hour without BDNF. You can see that less elaboration, less neurites growth after 27 hours, even after 22 hours. You can see that addition of BDNF causes many neurites growth, 39 hour or 43 hour. Like after 39 hour, you can see even more, neurites growth. In the control, you do not see such elaboration. So this happens because of the expression of neurotrophins, the BDNF, brain-derived neurotrophic factor in the cortical neurons. So here, showing that BDNF basically recruits its effector, like AKT, PKB, or ERK, or PLC gamma for the for the neuronal growth. So here the control, you have less neurites growth, but when they added BDNF after 5 hours, you can see that you have more neuronal growth. So you can see, after 5 hours. But when they, added these inhibitors for the, either for AKT or for PKB or for ERK, you can see that here you can see that they have added this 10 micromolar these inhibitors for the BDNF. You can see that you have less neuronal growth, or here they have added another inhibitors for the BDNF. These are showing that in the control you have BDNF with DMSO, you have more neurites growth. And when they added the inhibitors for this pathway, BDNF pathway, either for AKT or for PKB or for ERK or PLC gamma, you see reduction of the neuronal growth in all these situations, except cyclohexamide. Cyclohexamide actually inhibit the translation of the protein. So in that case, you do not see such effect like other inhibitors for the AKT or PKB or ERK or PLC gamma. Okay. So just 1 or 2 slides. So here showing AKT inhibition, showing PLC inhibition, then protein synthesis, the ERK inhibition. So these are the effector for the BDNF. Okay. So then the last slide kind of I'm talking about basically BDNF promotes dendrite growth in association with NMDA type glutamate receptor. Basically, you have activation of glutamate receptor and NMDA type. When the BDNF binds, you have activation of NMDA type glutamate receptor. Then you have neuronal growth. So here, this is the control untreated. You have growth of this in neurites, less growth of the neurites. Here they have blocked this. Here minus BDNF, here this side the plus BDNF. So here you are not using any BDNF, okay, only the control. And here in the control, they have blocked this NMDA receptor by using this drug, APV drug. You see more or similar neuronal growth. But when they added BDNF, here in the untreated, when you do not have the APV, you see after addition of BDNF, you have large number of neuronal growth. But when they block this receptor, the NMDA receptor, even they have BDNF, you do not see such neuronal growth. That means you need activation of NMDA receptor when you have BDNF signaling. So BDNF binds to the its receptor, then you have activation of NMDA receptor, then you have neuronal growth. Okay, so this experiment suggests that you need activation. So here kind of quantifying that in the control, here they have blocked the AMPA receptor, NMDA receptor, or the sodium channel. These are required for the activity, or L-type calcium channel. In all these situations, you do not see neuronal growth. But when they added BDNF to the control, you see large number of you have more neuronal growth. But when they added the BDNF along with this blocker for the neuronal activity, you do not see neuronal growth. So that means you need activity for the neuronal growth. Okay, so that suggests. So this is the last slide talking about how neurotrophic factor is important for neuronal growth. Okay, so that's all about for the lecture. If you have any questions, you can always Zoom with me or email me. So we'll talk more about this. You have 7 days to prepare your exam. I wish all the best for the exam. And don't worry about the questions. They are very simple. They all come from the slides. And I think you will do better than the previous exam. Thank you.